In November 2019, a pandemic changed the course of history. Like a scene in a horror film, COVID-19 was spreading like wildfire across the globe. Many lives were lost. Many felt utterly helpless. This tiny virus burned through the world, causing enormous devastation. When an apocalypse seemed inevitable, a lifeline emerged. Like a miracle, vaccines turned the tide of the situation and brought relief to the entire world. This newfound hope came from a technological innovation. The first mRNA vaccine for humans was developed in record-breaking time. The design and clinical trials took less than a year. It blunted the force of waves of infections. A group of dedicated scientists made the miracle possible. Among their ranks are Katalin Karikó and Weissman, who found a way to reduce mRNA immunogenicity, and Pieter Cullis, who designed lipid nanoparticles for the delivery of mRNA vaccines. The achievements of the three scientists saved countless lives worldwide and earned them the 2022 Tong Prize in Biopharmaceutical Science.
How the scientist is, you have so many problems you are trying to solve, technicals and others, and when you are in the laboratories, you are in full control, you can do that and that and that. And
with short hair, a sharp look, and a large pair of glasses, Katalin Karikó of the University of Pennsylvania is known as the mother of mRNA vaccine. Without her, mRNA vaccines would not have been in use so rapidly. For generations, vaccines have helped us prolong life, eradicate smallpox, fight polio, measles, and other deadly infectious diseases. They trained the immune system to recognize pathogens. As technology advanced, new vaccine development methods also emerged. Most traditional vaccines work by injecting inactivated or weakened viruses into the body to cause mild infection. In this way, the immune system learns how to identify and attack the invading pathogens. If the same pathogens appear again, the system will jump into action. mRNA vaccine is a kind of next-generation vaccine. Instead of pathogens, a manual is injected into the body, instructing cells to produce specific viral proteins to induce immune response and build up immunity. So, the body doesn't have to be exposed to the virus, but can still learn how to fight it, which ensures greater safety. The development of traditional vaccines takes years, even decades. It requires complicated viral culture purification and inactivation, followed by long clinical trials. By comparison, mRNAs can be synthesized once scientists learn the sequence of a virus. They don't need to collect virus samples themselves. The process takes only weeks. It is faster and cheaper. Clinical trials are much shorter too. If the virus mutates, scientists only need to change the instructions to create new vaccines. The flexibility is incredible. While the principle of mRNA vaccine may sound simple, the execution is fraught with difficulties. The story we want to tell you begins with Dr. Karikó. She was born in a small town in Hungary in 1955. Though her family wasn't wealthy, she was a carefree child.
Since my father was butcher, I was around and seeing what he was doing, and we had also a big garden. We had animals there, we had plants, and we had our own little garden with my sister, who's 3 years older, and we put seeds there and we watched what's happening. We didn't have television, we didn't have running water, but, we had electricity, let's say, and so we were very happy. High school, when I announced that I will be a scientist, although I haven't seen one, but I just figured out that this should be something exciting to go to work, and every day, that's how I imagined. Every day do something different and discovering something that nobody knows.
In 1953, scientists unveiled the double helix structure of DNA and revealed the secret of life. But it wasn't until 1961 when they finally understood that DNA has to be transcribed into single-stranded mRNA, and mRNA can then be translated into proteins. mRNA stands for messenger RNA. It tells cells what their tasks are, such as making proteins or carrying out repair work. It brings the instructions of DNA from the nucleus to the cytoplasm, where it binds with the protein factory, the ribosome, and the assembling of designated proteins starts. For decades, scientists have been dreaming of writing artificial instructions for mRNA to make the proteins they want and turn cells into the best defense weapons our bodies can have. A versatile drug manufacturer that has the potential of curing any disease known to mankind. This idea sounded promising, but it used to be just a theory, as scientists only began to know how mRNA worked. In the 1980s, manipulating mRNA was a basic research project that might never avail. The young Karikó had the same dream and was determined to make it come true. However, her life was anything but easy. After obtaining a PhD in biochemistry in Hungary in 1982, she joined the Biological Research Center to research RNA technology. She lost her job when the funding ran out. At 30, she decided to move to the U.S. to continue her work at Temple University.
Only permission we got to exchange $100. So coming with a family to the United States from Hungary with $100, it is just wouldn't be enough. And so what we did, we sold our car. We had a Russian-made car, and we sold it, and we exchanged in the black market. So I wrapped it up, and I cut up the back of my daughter's teddy bears, and I put the money, wrapped money in, and sewed it back, and I gave this teddy bear with 800 pounds in it to my daughter. She was 2.5 years old, and at the airport we kept our eye on Susan and the bear to make sure that, we don't lose all of this money.
They sold their car and left everything behind to cross the sea for a better life. As a foreigner, she had to adapt to a new life and a different language. Realizing her American dream was much harder than expected.
And coming to the United States, our living standard dropped because, we did not have a washing machine in our apartment. We had to rent, and we had to go to a basement to another buildings in the middle of the night and put the coins there to wash, to dry the clothes and so on. And so it was difficult. Laboratory was also kind of run down, so I had to fight with the cockroaches many times because they were running, and I was doing the experiment and run in the middle of the buffers, and so people think that everything is better here. It's not every place.
Difficulties abounded in both her life and career. She started working the next day she arrived in America and was in the lab almost every day. Back then, there was little enthusiasm for mRNA research, mainly because foreign mRNA can trigger innate immune responses, causing severe inflammation and side effects. mRNA is also extremely fragile. It is easily degradable and difficult to work with. Applying mRNA technology to humans seemed almost impossible. Facing a dead end and without a solution in sight, many scientists chose to throw in the towel.
The community who worked on this mRNA field, as you look at there, in the 1990s started 1 or 2 scientists, and they published, and they usually just publish 1 paper, and they are disappeared. Because, the RNA was, low amount of protein was made, and it was hard to get funding. I never get the funding for my work. We needed to generate more data. That's what we felt, because, the community, the other laboratories, not much data is coming out because they were not working on this.
In 1989, Dr. Karikó was hired by the University of Pennsylvania as an adjunct assistant professor. But her lab had to close due to a lack of funding. She had no fixed positions, no impressive publications. She was always writing proposals to apply for grants, but was always rejected. Eventually, the support from the university stopped. She was demoted, but she remained committed to studying mRNA and would not give up.
In 1995, I was also demoted from my position, but the work was so exciting, and we get better and better data, so that I thought that who cares about the position if the work is good? So I want to keep continuing. We never know what we will end up. Here we are. I never wanted to be in vaccine development. So in science, you don't know what you are doing, how at the end.
As an undervalued female scientist, Dr. Karikó was struggling on the margins of academia, trying to survive, not knowing what might happen. Her mRNA research was marked by failure, frustrations, and misfortunes. But this chance encounter may be an exception. During this trying period, Dr. Karikó met immunologist Drew Weissman at a photocopier. It changed their lives, and the fate of humanity was about to change as well. Dr. Weissman was another key figure behind the creation of mRNA vaccines. Born in Wisconsin, the United States, he has been fascinated by science since childhood.
I grew up in a middle-income family in a nice suburb. I developed type 1 diabetes when I was 5 years old, which radically changed my life and gave me more of an interest in science. And I never lost that interest, but you know, my family would call me stubborn. I call it persistence.
After getting a PhD in immunology, Dr. Weissman went on to be an outstanding physician scientist. Later on, he joined the National Institutes of Health and a team led by Dr. Anthony Fauci to do research on HIV. In 1997, he became an assistant professor at the University of Pennsylvania and set up his own lab to work on developing HIV vaccine. Major turning points in life often arrive serendipitously. Back in the era of photocopiers, at a university's photocopier, the two scientists met.
We met really at the copy machine where we copied the Xerox papers, and if it happens 2002, let's say, he comes 2002 here, we wouldn't meet because I downloaded everything digitally. But at that time, we still copied things. And he was a new guy on the floor, and then I noticed, because I was working already 10 years here, and then I introduced myself. I like to brag, and told him that I'm working with RNA. And he told me that he was coming from Anthony Fauci's lab, and he was working on HIV, and he wants to develop HIV vaccine. And so we talked and, agreed that I will make RNA and he will test out whether it will be a good vaccine. And that's how we started.
It's hard to remember that many years ago, but I'm sure that Katie started the conversation. Her RNA research wasn't going well, which is why she got demoted and why she didn't get grants. And that's because nobody understood that RNA was inflammatory. So I'm an immunologist, that's what I studied. So when the two of us came together, it made for kind of the perfect team to figure out how to make RNA work. I was a new faculty member. I got grants from the NIH, so I had money and I could fund all of our research. Never turn your back on somebody at the coffee machine.
A random conversation opened up brand new opportunities. Though Dr. Weissman was doing experiments with DNA at that time, he became interested in mRNA. Learning how to manipulate mRNA could lead to the creation of a new type of vaccine and infinite possibilities for medicine. They decided to work together, and their partnership has lasted more than 20 years. They worked shoulder to shoulder, often in difficult circumstances.
I don't think people realize that when we started working together, it was Katie and I working at a bench like this, side by side. It was just the two of us because we didn't have any grants, we didn't have any money to hire technicians. So Katie and I worked together for many years, side by side, We would then get the data and we would sit down and discuss what it meant and what we had to do next. But we often did that at 4 in the morning because neither of us sleep well. So we would be emailing each other at 4 in the morning, talking about the data and what to do next.
He's calm and I am more like excited. So our personality seems different, but when we looked at data, I mean, we cut each other words because we want to tell the others and the ideas what we got.
We have very different personalities. I always joked, or I used to joke to Katie back then, the way that she got from A to B was like this. And the way I did it is like this. We just had very different ways of thinking. But we both shared an interest in understanding, in discovering how things work, in having an open mind, and being able to take any data we get and try and understand what it meant. So that was the same in both of us.
Despite their different personalities, Dr. Karikó and Dr. Weissman made an excellent team because they had the same goal. They wanted to unravel the mysteries of mRNA and fight diseases. So, Dr. Karikó brought synthesized mRNA to Dr. Weissman's lab. They injected it into mice, but triggered harmful immune responses. Some mice got sick, and some died. Obviously, the immune system saw the synthetic mRNA as an intruder. But why was the mRNA produced in the body not targeted by immune cells? It took them several years to find the answer.
I was happy because I thought it would make a better vaccine, but I was discouraged because it would not work as a therapeutic. So we spent 7 years trying to figure out why it was inflammatory, what sensors, what proteins were recognizing it, and how to avoid the inflammation. And that was our big paper in 2005. We figured out how to modify the RNA so it was no longer inflammatory.
RNA is made up of 4 bases, A, C, G, and U. Their seminal 2005 paper showed that they were the first to prove foreign mRNA ignites the immune system because uridine is recognized by toll-like receptors, TLRs, on immune cells, thus provoking an attack. But natural RNAs have undergone several modifications within cells for them to travel safely in the body. Dr. Karikó and Dr. Weissman performed further tests and screenings, and finally reached an optimal solution in 2008. They simulated the cellular mechanism in modified mRNA by replacing uridine with pseudouridine. The disguise worked. The artificial mRNA was allowed into the body without causing any inflammation, and the proteins produced were 10 times the designated amount.
In this paper, we demonstrated that the uridine, one important component, one of the 4 building blocks of the mRNA, and all RNA actually, is causing inflammation. We identified the receptors, the sensors which activated by the uridine. Those were toll-like receptor 7 and toll-like receptor 8. And we also identified that if we change this uridine with a modified alternative, which is naturally also present in our body, then the RNA is not inflammatory. And later, we also went on, and in 2008, we demonstrated that when we changed this uridine to pseudouridine, and naturally occurring alternative, which is present in our body, that RNA is not only not inflammatory, but so much more protein is produced from such RNA. And we demonstrated in 2008 that even in animals, this was not inflammatory, and it translates so well and extended the translation.
The astounding results changed the rules of the game. Dr. Weissman was sure that it would shock the scientific community and their phones would not stop ringing. But that wasn't what happened. Their paper was rejected time and again. Even when finally published in the journal Immunity, it didn't attract much attention.
I don't know why. I think a lot of it was people had been burned by RNA. RNA is very difficult to work with, and there wasn't a lot of interest. It took about 3 or 4 years before basic science researchers produced something interesting.
We expected that people get excited and mentioning, you know, how our personality is so different. Drew is more withdrawing, but he was the one, he said, listen, you know, the telephone will ring. And of course, nobody called, not 2005, not 2008. People were not responding, you know, the community were not interested in it. But we had so much more to do, you know, to figure out in vivo studies, to figure out purification and many, many other things. So we were not sitting and watching the telephone to ring.
Their phones didn't ring off the hook. Their research was largely ignored. It was the greatest discovery they had ever made, but the world took no notice. However, they were not the ones who give up easily.
I would go to meetings, and I would talk to Tony Fauci, who I worked in his lab, and Gary Nabel, and other, you know, really high-station researchers. And they would listen, and they would say nice, and then they would say, why are you wasting your time with RNA? Why don't you do something worthwhile? And, you know, I would listen to them, but, you know, I saw the potential of RNA, and I saw, you know, we kept getting interesting results, and I just didn't want to give up.
Although people were yet to appreciate their valuable findings, they had found a way to prevent the severe side effects that emerge when foreign mRNA triggers immune responses. It was finally proven that nucleoside-modified mRNA can safely control how cells produce proteins. But there is one more crucial person we need to talk about. Without him, mRNA wouldn't be able to enter human cells and deliver its instructions. It is no easy feat to send mRNA into target cells. mRNA is very unstable and gets degraded by enzymes soon after it enters the body. If it can be enclosed in a membrane, it can be protected from harm and be released once inside the intended cells. That is where LNPs come in. Both mRNA and LNP technologies play a vital role in the development of mRNA vaccines. LNPs, shorthand for lipid nanoparticles, are tiny capsules used to deliver a variety of therapeutics. They are composed of lipids and can be loaded with the mRNA that encodes specific viral proteins, transporting it into cells intact. Dr. Pieter Cullis is a professor of biochemistry and molecular biology at the University of British Columbia in Canada. He is a pioneer in LNP technology and another linchpin in the development of mRNA vaccine. Many people may not know that he started as a student of physics but shifted to biochemistry for his postdoc studies.
My PhD in physics in 1972, a long, long time ago. And I realized I didn't really want to go on in physics, that the really interesting problems in physics seem to have been solved 100 years ago. And there seem to be lots more interesting problems outside physics, and particularly in the life sciences. There's many more problems than there are answers. And so I got a postdoctoral fellowship to go to Oxford to study biological membranes. I walked into the building, and the animal room was right next to the front door, and I thought, this is a bit different.
The transition was an eye-opening experience for him. He became interested in membranes that form a layer around cells. These membranes are composed of lipids. When
you got, say, 30 trillion cells in your body, and they're all surrounded by a membrane, and those membranes all have lipids in them. They kind of supply the structure membrane. So they're pretty important things. It is a sort of a bubble. You can think of it that way. Cells have to have an inside and an outside. Inside the cell, everything is under control, and so that it allows the cell to replicate and to make the materials it needs to make. Anyway, you need this bubble around it.
Inspired by the intricate workings of the human body, Dr. Cullis started to study membrane structure and its functions at the molecular level. He also worked with colleagues to develop a lipid delivery system. They first created liposomes that can carry small molecule anti-cancer drugs. He and his team then tried to develop an LNP system to deliver RNA and DNA-based drugs into the human body. It was a difficult task. Both DNA and RNA are large, negatively charged molecules, and they cannot be carried by liposomes at neutral pH. To stabilize negatively charged nucleic acid drugs, we need lipids with a positive charge. However, charged molecules can cause harm to cells. Through pH adjustment, Dr. Cullis managed to control positively charged LNPs, significantly reduce their toxicity, and successfully use them to deliver RNA therapeutics.
We often get 100,000, of these small cancer drugs inside one nanoparticle, 100 nanometer nanoparticle. When we're doing siRNA, which is much bigger, the small interfering RNA, so that's, then we can get maybe 1,000. A messenger RNA, which is much bigger than that, maybe we get 2 or 3 copies per nanoparticle. So the problem then becomes, because if these messenger RNAs are, if any bit of them is exposed to the outside, they're very likely to get broken down in the biological fluids.
To make LNPs, lipids and ethanol and RNA in aqueous buffer from separate inlets are combined through high-speed agitation. Both the mixing time and the size of LNPs can be precisely controlled. In 2014, Dr. Cullis' research caught the attention of Dr. Weissman and Dr. Karikó. They were developing an mRNA vaccine and were looking for a reliable delivery system. So they sought help from Dr. Cullis' team. Dr. Cullis never planned to develop vaccines, but a phone call changed his mind.
Then we got called up by Drew Weissman of the University of Pennsylvania, and Drew said, I'd like to try your systems as vaccine. We never thought about using them as a vaccine. But what he wanted to do was to encapsulate messenger RNA that coded for a protein that's on the surface of a virus and see whether or not there was a good. After not hearing from this collaborator for I think a month or 2, whatever it was, they eventually started to talk, and the guy who was doing this work said, well, I didn't want to get back to you because the results I saw, first of all, were too good. And he didn't trust them, so he had to do it again. And it still worked the second time.
Amazed by what LNP technology can do, the 3 scientists started collaborating, using LNPs to package mRNA flu vaccines. mRNA vaccine made its debut after undergoing small-scale clinical trials. That was the reason why, when the pandemic broke out, this platform had matured enough to be applied to develop COVID vaccine right away. Having ignored it for years, biopharmaceutical companies finally embraced this research. Moderna in Massachusetts and BioNTech in Germany both began to develop mRNA vaccines. Dr. Karikó joined BioNTech in 2013 as its vice president and a member of its RNA protein replacement therapies team. It was the first stable job she got in over 30 years. BioNTech would later work with American pharmaceutical giant Pfizer to develop an mRNA flu vaccine. Shortly before the clinical trials began, the COVID-19 pandemic hit. At the end of 2019, when people were celebrating the New Year, a mysterious virus started to spread in Wuhan, China. Cases of pneumonia began to appear in the Huanan seafood market. Patients suffered from a cough, high fevers, lung infections, and shortness of breath. These symptoms can deteriorate quickly and cause death. Our immune system became powerless in the face of it. The medical community was in utter shock. Infections and deaths soared globally. Many medical systems were overwhelmed and unable to cope. An unknown disease plunged the world into a panic. With the virus spreading rampantly, countries around the world were desperate to find cures. A race between the vaccine and the virus began. Within 2 months of the outbreak, the genomic sequencing of SARS-CoV-2 was published. Relevant data were integrated swiftly. BioNTech and Moderna then started to develop COVID vaccines. The design was completed within weeks. Clinical trials soon followed. Both companies adopted the same ingenious mechanism. A safe and stable vaccine containing viral mRNA sequences instructs cells to synthesize viral spike proteins, which in turn induce effective immune responses. Dr. Cullis' team contributed by developing LNPs for the vaccine. It was a matter of life and death, and the clock was ticking. A long period of darkness finally gave way to a ray of light. In November 2020, Dr. Karikó received a call.
Ugur Zahin called me up and wanted to know whether I am alone. And then I walked into the other room and I said, yeah, I am alone. And he told me that the data is in and, you know, how effective the vaccine is. And so that was a short phone call. And then I went back and I told my husband that the vaccine worked. I ate a box of chocolate-covered peanut, which is actually was introduced here in Philadelphia. They called Gruber's.
My family was actually mad at me for what I was doing. They were sitting by the TV waiting for the results. I was sitting at my computer working on the next thing. I think I was working on a pan-coronavirus vaccine. I knew from the phase 1 results that it was going to work because the antibody responses were so powerful. So I was sure it was going to work. I was hoping for 100, but it's probably asking for too much. I would have been very depressed if it was only 50% protection, because every animal model we had 100% protection.
I was amazed. You know, I was hoping that it was going to work. I mean, maybe 70%. You know, yeah, but 95%. I mean, I don't think anybody could quite imagine that happening. So that was incredible. No, I had a couple of glasses of scotch to celebrate. So it's,
The 3 scientists were overjoyed at the vaccine's 95% efficacy, but they had foreseen its success long ago. The mRNA COVID vaccine was created in 11 months, much shorter than the 4 years it took to develop the former record holder, the MMR vaccine. The vaccine is formulated with lipid nanoparticles that encapsulate artificially nucleoside-modified mRNA. Once entering the body, it directs cells to engineer harmless spike proteins and prompts the immune system to mount a defense. When the vaccine rollout began at the end of 2020, Dr. Karikó and Dr. Weissman received the shot alongside other medical workers. Years of hard work paid off. At the moment of this injection, they felt nothing but happiness.
It was 2020, December 18, I remember that, before Christmas, and then already the healthcare workers lining up in every 6 feet in the hallway to get the vaccine in another room. And then when the chairman said that, you know, these people were responsible for the vaccine, something he said like that, and the people on the hallway started to clap.
1, 2, 3. You will not believe how happy I am. Yay!
You know, even right now I'm talking, you know, my voice just chuckled, because it was realizing that those people, you know, those went to work every day in 2020 and risking their life, because they were, you know, went there and taking care of the patient, and they couldn't go home, or they were, you know, risking infecting their own family. And so they were the real heroes. And feeling the relief that now there is something to protect them was important.
They wanted initially to have me as the first person to get the vaccine, and me alone. And I said, no, first, Katie had to be there with me because we worked on this together and we needed to get it together. And then we needed to be after the first line workers, after the ER doctors and nurses and staff, because they're the ones that are doing the hard work. We're sitting in a nice, comfortable lab. They're sitting in the emergency room treating thousands and thousands of people who are very sick and not doing well. I think vindication is probably the right word, because I had spent 25 years working on something, and to suddenly have it being put in my arm and everybody else's arm was a sign that, yeah, it worked. We were right. We spent 25 years, and it was a good thing.
Dr. Cullis recalls his conversation with the nurse when rolling up his sleeve to get the first COVID shot.
Yeah, so I went and lined up with everybody else, and when I got to the, where the nurse was there, with the needle, I said, well, I had something to do with with how that vaccine got made, and she clearly didn't believe me. She gave me a look that was like I came from outer space.
Cometh the hour, cometh the man. Because of a once-in-a-lifetime pandemic, 3 previously unknown scientists were catapulted to fame. This time, no one ignored their achievements. International awards kept coming. Media coverage never waned, but they found the sudden stardom a bit hard to get used to.
I'm a quiet guy. I tell people nothing personal, but I'd much rather be in my lab working right now than talking to people.
I don't feel, that unique. I feel that so many other scientists like me, I was just working, doing my best, and there are so many scientists like me.
Dr. Karikó's life took yet another interesting twist. Remember her 2-year-old daughter who was entrusted with a teddy bear filled with cash? She grew up to be a professional rower and won gold medals in 2 Olympic Games. Dr. Karikó used to be known as the mother of an Olympic champion, but things have now changed.
This is when I work here at the University of Pennsylvania. They never said that, meet Kati, when they introduced me, meet Kati, she works with RNA. Who cares? They told, did you know Kati's daughter is Olympic champion? That was I was the mother of a champion. That was I was famous for. So now that things changed, and now she's bragging about that her mother is me.
Her persistence and talent have rubbed off on her daughter. Her family's unwavering support has allowed her to focus on her work in the lab. When asked about young people's lack of interest in a career in science, she encourages them to always look beneath the surface.
If they look at me outside, what I was doing, they can see I was a failure, no? I mean, I was working, I was kicked out, demoted, kicked out again, so on, and then my salary, was no worse. So it was like a technician's. So they can see it's not rewarded financially. She was not promoted, so even, the prestige is nothing. You are in a control. You are in the laboratory, the equipments are there, the gel apparatus. And then when in the evening, I was reading an article, and then oh my God, maybe that's the answer to my question. And I read, oh, I can do it. And next day I'm coming and I can do it. These empowering things that they have to feel, that they are in control. And not focus on so much what they cannot control. Who will be promoted, who will decide on your salary, and so on. And that's what, we try to convey and tell the young one, that you have to focus on what you can change and not what you cannot.
During the interview, Dr. Weissman pointed out that instead of continuously updating the vaccine to target COVID-19 variants, a pan-coronavirus vaccine should be developed to effectively counter any potential future mutations. It may sound like another pie-in-the-sky idea, but the success of mRNA vaccines has fueled the public's expectations.
We started working on a pan-coronavirus vaccine, and the idea was that it would be useful for any coronavirus that crossed over from bats. And there have been 3 in the past 20 years, so there's going to be more. But it would also be useful against any variants that appeared. We've developed one for it's called the 2B family. So that's the original SARS and COVID-19. And it protects against both of those. It protects against the variants, and it protects against related bat coronaviruses that have the potential to cross over. So we've made a vaccine that should work against every variant. We're now working on adding in the MERS family of viruses and the common cold viruses. So we have a vaccine that protects against every coronavirus.
He also plans to use mRNA vaccines to tackle diseases such as the flu and AIDS. There is always research to do, and his lab is always busy.
They're affected by the humidity, by the temperature in the room, just every silly thing you can imagine, the phase of the moon. Who knows?
Okay.
Circumstances have changed. The world has come a long way. Now Dr. Weissman no longer has to worry about funding. He can press ahead with the plans he has made. His lab now offers a summer program for high school and university students interested in science. His reputation has attracted many young people.
Yeah, when I told them, they were just incredibly excited that I was able to get this opportunity. And I had them Google Drew Weissman's name to see what he had done. He was very helpful in trying to interpret the data and also answer my questions and the broader applications of where this can go.
It felt surreal in a way because obviously we hear so much about this lab on the news and everybody knows who Dr. Weissman is. So being able to have an opportunity to work in this lab is definitely beyond my expectations. Again, knowing that you're working on something that's so current and is actually physically making a difference in the world that we live in today is a crazy feeling.
The same thing also happened to Dr. Cullis. Like a magnet, LNP technology keeps drawing talent to Vancouver to take part in one of the most talked about research of the day. With a constant flow of funding, Dr. Cullis's lab has all the resources it needs. Young researchers there are bursting with enthusiasm and have faith in what's to come.
他给了我很多空间去试不同的东西，因为有时候就是我们还是有一个大方向，但是他就很乐意给我去试一些新的东西。mRNA 它的研究就有很多就是可能性，我们就是对这个方向很有信心，就是因为它可以做的东西很多。
The techniques Dr. Cullis devised were essential to the use of LNPs approved or authorized for emergency use by the FDA. He has published more than 300 papers, holds more than 40 patents, and founded more than 10 companies. For him, the business world has more to offer than academia.
If we can get the company started with some really good people in it and then continue the support from academic resources, they tend to be quite successful. You have to prove yourself individually. You have to prove that you have an idea. You write a paper, you get it published. The system is that's the way it operates. It doesn't really operate on a big team. Companies are, if you want to get something done, you have to have a team, and they all have to be working in the same direction. And so it's very productive.
He has high hopes for the promises the combination of mRNA and LNP technologies holds.
The potential is there for these lipid nanoparticle messenger RNA therapeutics to be useful for almost every disease humans have. If a child is born now and is and there's lots of rare diseases that children can suffer from. Say they're not making a particular protein in the liver that is say vital for blood clotting. Then we can make the messenger RNA that will code for that protein and put that in a lipid nanoparticle. We can do that all within a matter of a month or two. And that will be a medicine that will save that child. Once you know what protein is involved in a disease, that you can then start to treat it very directly, but also you can do it very rapidly. It doesn't take 15 years and $1 billion, which is kind of the situation for other drugs. These can be developed extraordinarily quickly and relatively cheaply. I mean, $400,000, but you can have a drug that's very specifically made for you.
For the biopharmaceutical industry, it is a revolutionary change. What scientists have developed was not only a vaccine, but also a module capable of making various vaccines and drugs. The mRNA vaccine platform allows the synthesis of designated mRNA, which prompts our cells to produce antigens and therapeutic proteins. Dr. Cullis believes that in the next 10 years, up to 40% of the new drugs will be made this way. The possibilities are limitless. Science is full of surprises. Who could have predicted these developments?
Science is not really very linear. You don't go in a very logical way that you can see 2 years or 5 years or whatever ahead. It goes all over the place. And this is a great example. So most of our efforts in biochemistry have been focused on proteins and understanding how proteins are made and what their structure is. But it's equally true to say if you don't have the lipids there, well, nothing's going to work. For people that thought lipids were not that important, well, I think we've shown them wrong. So.
The tireless efforts of these three scientists have prevented countless tragedies. Though the battle between humans and viruses still rages on, the advent of COVID-19 vaccines has ushered in an era of mRNA, LNP therapies, and more medical wonders are yet to come.